Okay, so welcome to this next video in which we are discussing tertiary lymphoid organogenesis. Okay, so before we just discuss what tertiary lymphoid organogenesis is, I'm just making sure that we all know what the immune response is. So, we've discussed the acute inflammatory response, which is also called the innate immune response. We're now discussing the adaptive immune response, and I've separated the adaptive immune response into three separate responses. One to where you have an extracellular pathogen, and then two where you have intracellular pathogens. One where it's living within the cytoplasm of the cell, and the other where it's living within a phagosome within the cell. Okay, so let's begin with the humoral adaptive immune response. Okay, so what will happen is that a professional antigen-presenting cell will phagocytose the extracellular pathogen. Okay, so it starts with what's known as a professional antigen-presenting cell, and there are two main examples of professional antigen-presenting cells, which are the dendritic cell is the main example, and then the macrophage. Now there is a third, which we'll come on to in a moment, which is B cells, but B cells aren't usually going to actually initiate the humoral immune response. So the professional antigen-presenting cells which usually actually initiate the humoral adaptive immune response will be dendritic cells, but macrophages are also capable of doing this. Now we'll just abbreviate this to APC for antigen-presenting cell. Okay, so the professional antigen-presenting cell is going to phagocytose the extracellular pathogen and take it into a phagosome, and then in that phagosome it's going to break the pathogen down, and it's going to break down all sorts of antigens that are associated with the pathogen. Okay, so these might be antigens on the surface of the pathogen, for instance. Okay, now an antigen is just any protein which is capable of having an antibody made against it. Okay, so you cut these antigens up into fragments and you put them on major histocompatibility complex class 2. So this is really important to understand. Whenever you get an antigen from a phagosome, basically, so when the antigen comes from a phagosome within you, so these antigen-presenting cells have got these antigens from the phagosome where the microbe was, then the antigen will be, the antigen fragment will be presented on MHC class 2, standing for Major Histocompatibility Complex class 2. Okay, this professional antigen-presenting cell will then go to the lymph nodes, and in the lymph nodes there are naive CD4 positive T cells, and because it is presenting the antigen fragment on MHC class 2, it has to present it to a naive CD4 positive T cell, because CD4 is a protein on the surface of the T cell which binds to MHC class 2. Okay, now there are a huge number of naive CD4 positive T cells within the lymph node, and they all have slightly different specificities. So they'll all have a T cell receptor on their surface which will be specific to a certain antigen fragment. Okay, so these antigen presenting cells will take a long time to find a T cell that has a perfectly good match, basically, that has a T cell receptor which will bind to the antigen fragment that is on the MHC class 2 molecule. However, when the antigen presenting cell does find a naive CD4 positive T cell with a complementary T cell receptor, then what will happen is that naive CD4 positive T cell will be activated and it will undergo differentiation, and it will differentiate into what's known as a T helper 0 cell. Okay, so T helper 0 cell, and these are often abbreviated to T, and then you put an H at the bottom as a subscript and then a 0, like so. Okay, and then what happens is that this cell undergoes proliferation, so you end up with many T helper 0 cells. Okay, so you go from having just one T helper 0 cell to having absolutely loads of the things. So here we have a whole population of T helper 0 cells here. So 3 I've drawn, but of course you'll make thousands if not millions of the things. Okay, so what happens next? Well, next, something happens that is not well understood. Somehow, these T helper naught cells decide to differentiate into T helper 2 cells. Okay? And this is what's not understood, because these T helper naught cells can differentiate into a different type of T helper cell. In fact, there are many different types of T helper cell that we now know about, which they can differentiate into. We'll only discuss 2 types in this video, T helper 2 and T helper 1 cells, which are the ones which most is known about. Okay, so what happens is that these cells start to differentiate into T helper 2 cells, okay, and these cells are going to help destroy extracellular pathogens. So they are part of the humoral immune response. So something tells these T helper 0 cells that we are dealing with an extracellular pathogen here, and they need to differentiate into T helper 2 cells. Now, one of the molecules that seems to be extremely important in this is interleukin 4, but we don't know where the interleukin 4 comes from, and we don't know how whichever cell releases the interleukin 4 knows that the pathogen is an extracellular pathogen. Okay, so that's still a mystery, basically. Then what's going to happen is these T helper 2 cells are going to help out with producing a B cell, which is going to make antibody against the antigen. Okay, so now what's going to happen concurrently is that you're going to get activation of B cells. So, basically, when the pathogen has infected your tissue, what will happen is you've brought in all of those neutrophils, okay? Now, neutrophils, when they phagocytose the pathogen and break it down, they do quite a poor job, and they start chucking out antibodies that are associated with the pathogen, okay? So they basically break up the pathogen, but they don't sort of finish the job and actually break down the antigens. Instead, they release the antigens into the extracellular fluid. Now, this is actually helpful because it plays a role in the recruitment of the B cells, or the activation of the B cells. Okay, so if you're chucking these antibodies into, these antigens into the interstitial fluid, then they're going to drain in the lymph vessels to the lymph nodes. And there, what will happen is they will activate B cells, or at least begin the activation process for B cells. So, in the lymph nodes, I'll draw this concurrently occurring to here. So in the lymph nodes, you have many different B cells. Okay, so you have a huge number of B cells, and they all have different B cell receptors. Okay, so this is a very important point to understand. Basically, every B cell will have a different B cell receptor, and some B cells will have B cell receptors that are just right for this antigen, basically, and bind really well to the antigen. So what will happen is we're going to be draining all of this lymph from the inflamed tissue, and this will be full of these antigens. Now, these antigens will be dripping through the lymph node, and they drip through the drip past the lymphocytes, okay? So these B cells will be exposed to the antigen, and eventually what will happen is this antigen will just happen to bind to a B cell receptor really well, and that B cell will now begin the process of activation. So what will happen is the B cell will internalize the receptor into a endosome, into an endocytic vesicle, okay? And then, when you have endocytosed an antigen like that, just like in the case of a phagosome, if it's in an endocytic vesicle, again, the antigen will be broken down, and then its fragments will be exposed on MHC class 2. So again, the B cell is going to express the antigen fragment on MHC class 2. It's then going to present it to this T helper 2 cell, which is armed against that antigen fragment, and the T helper 2 cell delivers a stimulatory signal to this B cell. Okay, now it would be lovely and simple if the B cell now just divided and made cells which are going to make antibodies. However, it doesn't. There's a slightly more complicated process that's going to happen. So the B cell is now going to divide, that's true, but it's more complicated. Okay, so it's going to produce a whole colony of B cells. Okay, so here is this whole colony of B cells, all 3 of them. No, it'll make far more than that. And basically what happens is a process known as somatic hypermutation. Okay, and somatic hypermutation involves a mutation occurring in the B cell receptor. So basically all of these B cells are going to undergo slight mutations in their B cell receptor. So, let me repeat it. We started off with this B cell, which had a B cell receptor which was complementary for the antigen. Okay? The antigen bound to that B cell receptor and was internalized, and the B cell ended up presenting fragments of the antigen on MHC class 2. Okay? It then presented this to the T helper 2 cells, which we've activated and are also in the lymph node. Okay? And those T helper 2 cells gave signals to the B cell for it to divide. But when it divides, it produces a whole bunch more B cells, and all of the offspring undergo mutations in their B cell receptor, so they all have slightly different B cell receptors. I mean, they're all based on the original B cell receptor of this original B cell, but they're all slightly different. Now, what's going to happen is a process known as affinity maturation, because you're going to set up a selection process, basically. Because basically some of these B cells here, and I'll put B's in them to indicate that they're B cells, some of them will have undergone mutations that mean that their B cell receptor is now better at binding to the antigen, whereas some will have undergone mutations that now mean that their B cell receptor is less good at binding to the antigen. Okay, so those B cells which have undergone mutations which mean that their B cell receptor is better at binding to the antigen, they are more likely to bind antigen, basically. So they're going to get antigen, whereas the B cells which have undergone mutations which mean that their B cell receptors are less good at binding antigen, they won't get antigen. Okay, and now to survive, basically, you need to get antigen. Okay, so that's the game. We have a certain amount of antigen. We have a huge, great population of B cells. Which of the B cells are going to get the antigen? It's the ones which have better B cell receptors. Okay, now the ones which get the antigen, what they do is they go through this process again. The antigen binds to the B cell receptor, and they internalize the receptor, break down the antigen, present the fragment on MHC class 2 to a T helper 2 cell. The T helper 2 cell gives them signals which make them survive and also divide. So the good ones will survive and divide, whereas the bad ones, the ones which have lower affinity B cell receptors, they will just undergo apoptosis. When they don't get T cell help, as it's called, so this process is known as T cell help, they will undergo apoptosis. Now, as you can see, this is a selection process, because it will gradually mean that your population evolves and you get B cells that are having better and better affinity B cell receptors for the antigen, because only those ones will survive. The ones which will survive are the ones which have the greatest affinity B cell receptor, and the ones which can't compete, which have less good B cell receptors, they'll gradually get picked off, they'll die. Okay, and when you get, if you got one that's originally really, really good, but then you're making ones which are even better, then even this one that was really good will eventually get selected out, basically. It will die, because if it can't compete against the ones which are even now better, then it's not going to get antigen, and therefore it won't get T cell help, and therefore it will commit apoptosis. Okay, so what is happening within the lymph node is that you're gradually producing a B cell population which has fantastically good B cell receptors for binding to this antigen. And this process is known as affinity maturation, and you should be very wary because there is a lot of misinformation about this process on the internet. So if you read Wikipedia pages about the adaptive immune system, you will read things where they're telling you what affinity maturation is, and you will be reading it thinking this is absolutely wrong, basically. Some Wikipedia pages say that affinity maturation is about the selection of T helper 2 versus T helper 1 responses, which is not right. It's this process of the B cells getting gradually better and better B cell receptors. Okay, now this process could continue on forever. You could just build a population of B cells with better and better B cell receptors. However, eventually what happens is the B cells get fed up, and one of these B cells will decide to come out of this process, and what it will become is a plasminoblast. Okay, so it will become a plasminoblast. Okay, and this plasminoblast will then divide and divide and divide and produce plasma cells. Okay, so it will produce many plasma cells. Now these plasma cells are finally terminally differentiated cells. Okay, and they will go into the bloodstream, and that should be a plasma cells, because one B cell will become one plasminoblast. The plasminoblast will divide and divide and divide and divide to produce a whole population of identical plasminoblasts, which all have the exact same B cell receptor, and then they'll all differentiate into plasma cells. And these plasma cells will then start chucking out antibody. Okay, so these will go into the blood and they'll produce antibody. And antibody is effectively just a soluble version of the B cell receptor. Okay, so it's a protein which can bind to this antigen with really high affinity now because of this affinity maturation process. Okay, now what is antibody going to do? Well, it's going to bind to the antigen on the surface of cells. Okay, so it'll bind to antigen on the surface of the cell. So let me draw a little microbe cell here. Then we've got the antigen here, and then we've got the antibody bound on top. And antibodies have these characteristic Y-shaped structures, where you have 2 heavy chains and 2 light chains. Okay, so the heavy chains I'll highlight in orange here, and the light chains I'll highlight in purple. Okay, and this will bind to the antigen, and what that leads to is it leads to opsonization. Okay, so opsonization is a fantastic word which means increasing the likelihood that this microbe cell will be phagocytosed. So basically, in order for a phagocyte such as a neutrophil or macrophage to phagocytose this microbe, it has to bind first to something on the surface of the microbe. And antibodies are one of the things that it will bind to. So if you're coated in antibodies, you're far more likely to be phagocytosed. Okay. The other thing this can lead to is it can activate complement cascades on the surface of the microbe. Okay, and these complement cascades can lead to the formation of membrane attack complexes, which destroy the integrity of the cell membrane, allow water to move into the cell, and cause the cell to burst, basically, by osmotic lysis. In addition, some antibodies will not be launched against antigens which are on the surface of the microbe. Instead, some antibodies might be against soluble antigens. Okay, so maybe the antigen was being released by the microbe. Okay, and this antigen that we've formed this antibody against, maybe it's a toxin, maybe it's some horrible molecule which is attacking our cells. Well, now if we bind a great big antibody onto it, then the antibody will neutralize the toxin. So the final role of antibodies is in neutralization of toxins. Okay, and that can also protect the metaorganism. Okay, so that is how you launch an adaptive immune response against an extracellular pathogen. And you'll be pleased to know that the humoral adaptive immune response is the most complicated of them. The other adaptive immune responses are far more straightforward. It's this interaction between the T helper 2 cells and the B cells which is confusing and takes the time to explain. So the other two will be very quick, and then we'll get on to tertiary lymphoid organogenesis.